Lakmini, you can have the session now.
Thank you, Pimi, and good evening, everyone. And for some of you, maybe good morning. And I want to thank Kushani and the organizing team for inviting me this year also. So in this, can you see my screen? Sorry.
Yes, yeah, we can.
You can, okay. So in this talk, I want to explore an emerging area of research, epigenetic modulation of the NLRP6 inflammasome and its potential as a therapeutic approach for reducing necroptosis-driven gastrointestinal dysfunction, especially in the context of HIV and SIV infection. So first, I will explain about intestinal barrier disruption. This image shows how damaged the intestinal epithelial barrier is leading to inflammatory-driven comorbidities. When the intestinal barrier is compromised, it leads to microbial translocation, which is proven in humans. And this means harmful bacteria and their components penetrate the gut lining and enter the bloodstream, triggering systemic inflammation. So this chronic inflammation contributes to the development of conditions like liver disease, diabetes, obesity, hyperlipidemia, and also cardiovascular and also neurological diseases. And this epithelial barrier disruption, dysbiosis, and microbial translocation persist in people living with HIV, regardless of their antiretroviral therapy, which is I'll call ART from now on. So even though ART drugs suppress plasma viremia, which is the viral loads in the plasma, these drugs cannot fully restore the disrupted intestinal epithelial barrier or completely recover the chronic immune activation in the intestinal epithelium. So people living with HIV have an increased risk for all these non-AIDS related comorbidities. So inflammasome activation play a very important role in intestinal barrier because it triggers the release of pro-inflammatory cytokines and also affects the barrier integrity. So inflammasomes are multi-protein complexes that play a big role in the innate immune system by triggering inflammatory responses. So they are key for recognizing pathogens and also endogenous danger signals, and then activate the pro-inflammatory cytokines. So while inflammasomes are essential for clearing infections, their dysregulation has been implicated in the pathogenesis of so many diseases. For example, heart disease, type 2 diabetes, cancer, and then inflammatory bowel disease in the intestine, and also neurodegenerative diseases, and also especially in infectious diseases like HIV and tuberculosis. So there are several types of inflammasomes, and each is activated by different stimuli. You can see the different types of inflammasomes in this blue circle, and the stimuli are on this outer circle. So this stimuli can be microbial pathogens and sometimes RNA or DNA viruses, and there are other examples here as you see. So each inflammasome has unique triggers and roles in various diseases, making them critical therapeutic targets. So I'm focusing on NLRP6 inflammasome here. So although its activators are not well defined yet, microbial metabolites and also recently some studies have found that double-stranded RNA stimulates the NLRP6 inflammasome. So NLRP6 is usually linked to maintaining intestinal homeostasis and protecting against gut inflammation and in healthy people, but its dysfunction causes chronic inflammation and metabolic disorders. So when you look at the inflammasome regulation, epigenetic mechanisms play an important role at the transcriptional level. So these epigenetic changes can affect how genes involved in inflammasome activation and can be turned on or off in response to environmental or pathological conditions. There are three main epigenetic mechanisms involved in regulating inflammasome activity. One is DNA methylation, and second one is histone modification, and also via non-coding RNAs. It could be either long non-coding RNAs or microRNAs. So microRNAs are usually 20 nucleotides long. So in this talk, I will be focusing only on DNA methylation as the epigenetic regulator. So what is DNA methylation? DNA methylation is an epigenetic mechanism involving the addition of a methyl group to DNA at cytosine bases in cytosine phosphate guanine called CPG islands. This can change gene expression without changing the DNA sequence itself, and abnormal DNA methylation patterns are linked to many diseases. So, but however, specific drugs that directly target DNA methylation to control inflammasome activity are not widely established in the clinical practice yet. So most current therapies focus on general DNA methylation inhibitors that have broader effects on gene expression. These are some of the current drugs, but no drugs are currently approved, especially for targeting DNA methylation in inflammasome-related diseases. So since drug abuse is very common in people living with HIV, in 2006, my postdoctoral supervisor, Dr. Mahesh Mohan's lab, did this study to investigate the effect of phytocannabinoids, which are immunosuppressive on an immunosuppressive disease. The hypothesis was that this would lead to more severe immunosuppression and drive faster AIDS progression and death in people. But the opposite happened as SIV infected rhesus macaques. So SIV is the simian version of it's simian immunodeficiency virus. It's the primate version of HIV. So the SIV-infected rhesus macaque that received long-term low-dose phytocannabinoids, THC, therapy survived longer than their vehicle-treated counterparts. So this finding led us to study the therapeutic effects of low-dose cannabinoids during HIV infection. So we have proven that phytocannabinoids reduce intestinal inflammation, has epigenetic reduce intestinal inflammation, and also has epigenetic benefits in our previous studies. So next we wanted to see its effect on DNA methylation and how it might affect to reduce inflammation and related comorbidities. We used an SIV-infected rhesus macaques model, and the study design is shown here. We used three groups: THC SIV, THC treated and SIV infected, and then vehicle SIV and uninfected controls. And then we collected colon tissues from all three groups at necropsy time point. And then we performed reduced representation by sulfide sequence of the colon epithelium to look into DNA methylation profiles. So based on the findings from DNA methylation profiling, relative to control, both vehicle SIV and THC SIV rhesus macaques showed more differentially methylated CpG sites distributed throughout all chromosomes. We see more hypo and hypermethylation in THC group. So vehicle versus control is in red color, THC versus control is in blue, and vehicle versus THC is in green color here. And above 0 is hypermethylated and below 0 is hypomethylated. So relative to controls, THC SIV group also showed increased hypermethylation in all 3 genomic regions, which is the promoters, exons, and also introns compared to vehicle SIV group. And then using gene ontology enrichment analysis, we investigated the effect of the methylation in the promoter-associated CpG islands of protein coding genes on the biological processes in the colon epithelium. We saw that THC-SIV, in THC-SIV, NLRP6 inflammasome complex assembly was hypermethylated relative to control. So we decided to focus further on that finding. So this is the genome track of NLRP6, including the location of CpG island shown here in green color. And it spans 3 exons and 3 introns, including a promoter region. And differentially methylated sites in THC-SIV versus control are in blue color bars on top, and red bars show the sites in vehicle SIV versus control, and then green bars show the sites in vehicle SIV versus THC-SIV. And this box represents the promoter-associated CpG island. And you can clearly see that THC-SIV versus control has more differentially methylated sites in this promoter-associated CpG island compared to vehicle versus control. Since methylation changes alter gene expression, we next investigated NLRP6 protein expression levels in the colon of all 3 groups. NLRP6 staining intensity had a decreasing trend exclusively in THC-SIV relative to controls and vehicle SIV group. So here, this is the colon epithelium. So these long the areas you see, that's the epithelial regions. And the areas in between those epithelial regions are lamina propria cells. So in a similar pattern, NLRP6 protein expression was reduced in lamina propria cells, so the lamina propria cells are usually mainly immune cells. So it was reduced in THC-SIV group compared to vehicle and control. So this study was a cross-sectional study, and maybe that's why we didn't see a statistically significant difference. So for the next study, we actually used a longitudinal study design. Since we confirmed that HIV-SIV infection stimulates the NLRP6 in the intestine, next we wanted to see what are the downstream pathways associated with NLRP6. And previous studies have shown that NLRP6 is an activator of both pyroptosis and necroptosis program cell death pathways. So these are two completely different signaling pathways that involve different proteins. Pyroptosis is caspase-1 and gasdermin D dependent, and necroptosis involves MLKL and RIPK proteins. So we then looked at these components of necroptosis pathway. Our gene expression data from colon epithelium showed significant upregulation of RIPK1, RIPK3, and MLKL genes in vehicle SIV animals. And RIPK1 showed significant upregulation in THC group as well, but RIPK3 and MLKL expression was significantly increased only in vehicle SIV group. And RIPK3 phosphorylated on serine 227 and serine 199 plays an important role in the activation of MLKL by phosphorylating it on threonine 357 and threonine 358 to enable its loading onto the necrosome. And finally, HMGB1, this is a protein that increases inflammation associated with necroptotic cell death. And so this was significantly increased only in vehicle SIV and not in THC SIV group. So activated RIPK1 activates RIPK3 phosphorylation, which then autophosphorylates itself on serine 199 and serine 227. So we detected phosphorylated RIPK3 serine 199 significantly higher, highly expressed in both vehicle SIV and THC SIV compared to control group. And so consistent with the high MLKL gene expression data, protein expression of necroptosis marker phosphorylated MLKL 357 was significantly upregulated in intestinal epithelium of vehicle SIV animals. And THC group showed a significant increase compared to controls, but significant reduction compared to vehicle group. And then phosphorylated MLKL 358 also showed similar expression pattern as MLKL 357 in colon epithelium, but here it wasn't significant between vehicle versus THC animals. And finally, we looked at the HMGB1 protein expression. And this is very important because it's a danger-associated molecular pattern protein released upon prior necroptotic cell death. And it's an inhibitor of it's an initiator of secondary inflammatory responses in neighboring cells as well. Although it wasn't significant, it was markedly increased in vehicle SIV animal compared to control group and THC SIV group. So our findings, to summarize, our findings offer new insights into the epigenetic mechanisms driving gene expression in colon epithelial cells during chronic HIV/SIV infection, influencing inflammation. And these findings demonstrate for the first time the activation of necroptosis and its contribution to intestinal dysfunction in HIV/SIV infection. And this provides insights into how low-dose long-term phytocannabinoids potentially through epigenetic modulation of NLRP6 can reduce necroptotic cell death. So since the majority of this audience are from math background, I wanted to mention how mathematics can be applied to epigenetic regulation. So I don't know much about the math, but so I did a quick Google search, and these are what these are the ones I found. The first one is mathematical modeling can be used to identify DNA methylation dynamics. For example, how methylation levels changes over time based on enzymatic activity, and then model gene regulatory networks like interactions between transcription factors, DNA methylation, and gene expression, and model epigenetic interaction networks like interactions between DNA methylation, histone modifications, and also doing DNA methylation and disease progression models. So with that, I would like to thank our lab members and especially Tulane and SNPR CIVET team and our collaborators. And finally, NIH Schneider for funding the projects. And thank you all for listening, and I'm open for any questions.
Thank you, Dr. Lakmini. Any questions?